Well, in this lecture, we're going to be discussing the physiology of striated muscle. Your textbook goes into smooth muscle and cardiac muscle, but we will not be discussing that in this lecture. So we need to review a little bit of the anatomy, the gross anatomy, particularly this upper part here of the slide, before we get into the more important part from the physiological standpoint, which is the second half of this list of structures. So first of all, a muscle shown here in this diagram. We have a muscle that is made up of a group of fascicles. So we have the fascicle shown here. Fascicles themselves are a bundle of muscle fibers. Important thing to keep in mind is that the term muscle fiber refers to a muscle cell. So don't think of it as something that's bigger and more complicated than the cell. It's actually quite complicated enough. The muscle fibers are surrounded by connective tissue. So all of this white material you see here is connective tissue, which of course helps to support the muscle fibers. The term sarcolemma is used to refer to the plasma membrane of the muscle fiber. So it's just another term for the cell membrane and muscle fiber. The muscle fibers are multinucleated. There's a fair amount of synthetic activity going on within a muscle cell, muscle fiber. Therefore, we need numerous nuclei to drive that activity. The term sarcoplasm is used to refer to the cytoplasm or the intracellular fluid of the muscle cell. What we're going to really emphasize for the rest of this lecture is this next few points. We'll be going into them in some detail. A muscle cell contains many myofibrils. Myofibrils do not really show in this diagram. It makes an attempt to pull out a few, but you can't see it in any detail. We'll be talking about the myofibrils starting in the next slide. Also contains what's called the sarcoplasmic reticulum. I've already talked about the endoplasmic reticulum as a storage site for calcium when I talked about calcium and its role in signaling. Same thing holds here. It's just that we use a special term, sarcoplasmic reticulum, to refer to it, and it has the same function. It's a calcium storage. Calcium plays an incredibly important role in muscle contraction. We'll see that in a few more slides. Of course, for muscle contraction to occur, we need energy. So the muscle cells contain many, many mitochondria. Also, not showing at all in this diagram, we'll see it in a subsequent one, but what are called transverse tubules, or T-tubules. These actually couple the action potential that occurs on the membrane of the muscle cell to calcium release. So we can see those T-tubules here, and they traverse going crossways across the muscle. So the structure at the molecular level, this is what we're most interested in now, is at the molecular level. The fiber contains many fibrils. These are the little punctate spots shown here in greater magnitude. These fibrils give the muscle the striated appearance that gives it its name. So we have basically these layers that create a striation that looks like it goes crossways on the muscle. And again, it is these myofibrils, these proteins that are shown in this diagram that contribute to that. These are called the thick and thin filaments. The thick filaments shown here in gold, and the thin filaments shown here in this sort of purplish blue in the diagram. And it's the orderly arrangement of these filaments in what is first of all called a sarcomere that causes this striation. The orderly arrangement of the thick and thin filaments. The thin filaments are actin. They are the thin filament shown here in this sort of purplish blue in the diagram. And myosin are the thick filaments, which are the ones that are shown in the diagram as being sort of the gold or the tan color. These filaments form, as I said, what we call a sarcomere. Now, the sarcomere is the contractile unit. What we mean by that is that, of course, a whole muscle is made up of many muscle cells. The muscle cells themselves are made up of many sarcomeres, and as shown in this diagram here. We have one sarcomere, another one right next to it, yet another one beginning here, and this goes on back. Each of these is a sarcomere. So each of the sarcomere acts as a contractile unit, that is a machine that causes shortening of the muscle. And what we'll see is that the distance between these two so-called Z lines actually shortens when the muscle contracts. And it is because of molecular interactions between the contractile proteins, the actin and myosin, that causes this to happen. So the thin filament, or the thin myofilament, is actin. It is a contractile protein. It causes muscle contraction. We talked in the lecture on structure of proteins about the different levels of structure as well as the different types of structures that can be formed. Actin actually starts out as what's called G-actin, which is formed of these little globules, hence the G of actin. G-actin molecules bind together in this filament that is then called F-actin because it's a filament-like structure. The filaments together form a double helical strand. So we have the two F filaments. Here's one, and here's another, wrapping around each other. If you notice these little white spots, black spots on the G-actin, these are myosin binding sites. So these are the sites that are going to allow the actin to actually chemically bind to the myosin. It is going to cause a muscle contraction. Also shown here is tropomyosin. Tropomyosin is a regulatory protein. It regulates the ability of the actin to bind to the myosin. And it overlaps the binding sites for myosin on the actin. So as you see here, all of these myosin binding sites are actually obscured, overlapped by the tropomyosin, so that those myosin binding sites on the actin would not be able to bind the actin. Be able to bind to the myosin. So this is the regulation of the binding we're referring to here. Troponin is this large pink structure shown here. It's actually a complex of 3 proteins. It is also a regulatory protein. Troponin T binds to tropomyosin. So it's what actually anchors the troponin to the tropomyosin. Troponin I attaches to the actin and inhibits the contraction. So it's actually the part that is bound to the myosin binding sites at each of these places. And troponin C binds to calcium reversibly. If you keep in mind what their names are, troponin I inhibits contraction, troponin T binds to tropomyosin, and troponin C binds to calcium. The names of the 3 subcomponents help to tell you what they do. So calcium binding to the troponin is going to regulate the ability of the actin to bind to the calcium and therefore to cause muscle contraction. The thick filament, we're talking about this large gold or tan-colored structure here, is made of myosin. Myosin consists of both a tail, which is a long filamentous type of structure, and a globular head. The tail is just a way of bundling all the myosin together in this thick filament. The head is the really important part because it contains the actin binding site as well as an ATPase site that breaks down ATP, which is important for regulating the binding between the myosin and the actin. So calcium regulates actin's ability to bind to myosin. ATP regulates myosin's ability to bind to actin. So first of all, of course, it has the actin binding site, which is all important, and this nucleotide binding site for ATP, and also has an ATPase, the enzyme that breaks down ATP. So it's not only going to be binding to the ATP, but it's then going to be metabolizing it. And the heads are arranged in a spiral around the tails. You can sort of see that here, that heads are lining up, forming this spiral-like arrangement. In fact, it kind of looks like threads on a screw. And what might be more accurate is, since the lines I'm drawing are, of course, obscuring the other side, is to think of this as actually some sort of a coiled or spiral type of structure. And this is the spiral arrangement that we're referring to here. The myosin heads are going to form what we call cross bridges between the myosin filament and the actin. And those are a little hard to see in this diagram, but these are the little spots. Let me switch to a different color. It might show better. These are these little protuberances coming out from the myosin toward the actin. These are what we refer to as cross bridges because they bridge the space across the area between the thick and thick filaments. And we're going to refer to the binding of these heads to the actin as the sliding filament mechanism, or a cross bridge cycle, which is going to cause the muscle to shorten. Excuse me. We'll be talking about that more in a few slides. One other protein we have to talk about is titin. It anchors the thick filaments between the Z lines. So let me switch to a different color. We have our thick filament here. And if you notice at the end of it, there is this smaller tan structure. And I'm going to switch colors so it'll be easier to see. This smaller protein here. This is the titin we're referring to. And the Z line is basically where the actin comes together in this Z-shaped structure, hence the name Z line. The titin provides structural support. It actually helps to keep these thick filaments suspended here in space. Because notice that the actin molecules all attached to the Z line. The myosin, it doesn't. It actually is suspended in space by these little titin appendages on the end. The other thing is that titin is very elastic. It's like a rubber band. It can stretch and it recoils after it's been stretched. In your laboratory exercise, it's going to refer to a passive component of muscle tension, and a large amount of that is due to titin. Excuse me. So the sliding filament mechanism, well, it's what causes a muscle to contract. First of all, muscle contraction is the development of muscle tension. The tension causes the shortening of the muscle, and as we'll see, ultimately the lifting of a load. Sliding filament mechanism refers to the thick and thin filaments that overlap, and neither of them will shorten. Let me switch to a different color here. When you draw the lines down here through our Z lines, the lower sarcomere is shorter than the upper sarcomere. But neither the actin filaments nor the myosin filaments themselves shorten. They're the same length. That the two thick and thin filaments slide past each other. And this is what we refer to as the sliding filament mechanism. And when this happens, the sarcomere shortens. And of course, when a muscle shortens, it generates force. This shortening is due to the cyclical formation and breaking of the myosin head binding to the actin here, what we call the cross bridge cycle. So we're going to look at this in some detail. And I'm going to probably switch back and forth between this slide and this figure. So first of all, in the cross bridge cycle, we're referring to the myosin heads and the fact that they undergo a conformational change. That is, as you see in this slide, the myosin head is going to have one angle here versus another angle here with respect to the myosin tail. So this is the conformational change we're referring to. And if you remember back when we had the lecture on proteins, that what makes proteins important is that when they bind to other substances, whether or not the other substance is a protein, when they bind to a ligand, they change shape, they change their conformation, and that change in conformation generally is what gives them whatever their function is. So the conformational change is a swiveling back and forth of the myosin head. And it's swiveling back and forth between two conformations, what we call the high energy conformation and the low energy conformation. The high energy conformation has a high affinity for actin. So this is when the myosin head is going to be bound to the actin molecule. And at that time, it also is bound to ADP and phosphate. Bound to ADP and phosphate. The low energy conformation has a low affinity for actin. So in that case, it ceases to bind to the actin. And at that same time, it is bound to ATP. It is also bound to ATP. Now keep in mind that the head contains an ATPase, so it's bound to the ATP. But of course, ATP will break down into ADP plus a phosphate. So on the diagram we see here, we have the ATP binding and then dissociating into ADP and phosphate. So this whole cycle relies on the hydrolysis or the breakdown of ATP. It produces what we call the power stroke. If you're out in a rowboat, rowing a boat, when you put your oars down into the water and then pull against it, it's the power stroke. In this case, it's causing the myosin head to move. So let's go back to our diagram. We have binding to the actin here, and notice the angle of the head. It stays bound to the actin, but notice that that angle has rotated about 90 degrees. So this is the power stroke. And of course, when it happens, it propels the thin filament, that is the actin, toward the center of the muscle. So when it goes from this angle to this angle, it propels the actin toward the center of the sarcomere. Then the thick and thin filaments detach, that is the myosin head releases. Myosin head then will release from the actin, as of course it's going from its high affinity to its low affinity site, or state, I should say, conformation. And the myosin head returns to its initial position. So I've more or less walked you through this cycle in switching back and forth between the two slides. There's one additional thing I want to point out. Step 3 here, which they refer to as rigor, which is when myosin is in the low energy state. So that goes back here. This is what we refer to as rigor. Rigor sounds like the word rigid, and it's when the muscle is tense. It's when it is rigid. It's when it's contracted. But you've probably also heard of the term rigor mortis, which refers to the rigor of death. Cadavers, deceased individuals, the muscle will stiffen over several hours and stays stiffened for quite some time. Well, in order for the muscle to go from this rigor state, where you have the actin and myosin cross bridge forming, ATP, when it binds to the myosin head, is going to permit the dissociation of the myosin from the actin, which is going to allow the muscle to relax. Getting ATP to the muscle, of course, is going to require a living organism with blood flow, bringing ATP to the muscle. So that is why rigor mortis occurs in a deceased individual, is in the absence of ATP, the muscle cannot relax. Sort of counterintuitive. You might think of ATP as causing contraction, not causing relaxation, but in fact it does cause relaxation. I've talked about this coupling or binding or cycling of the myosin heads. It happens asynchronously. you can't have go back here. We can't have all of the different individual myosin heads bound at the same time onto the actin. So what you have, if you take a look at the muscle fibers in cross-section, is this orderly matrix of myosin and actin fibers where each one is surrounded by the other in a very uniform way, so that you can continuously generate force in the muscle by having different myosin molecules either binding or dissociating from the actin molecules. So you never have complete loss of contact between the thick and thin filaments. There is always some cross bridges intact. Some, but not all, of the myosin heads are bound to actin at any given time. And it's this asynchronous nature, the fact that they're not all bound at the same time, that allows a smooth, fluid contraction that allows the continuous generation of force. I mentioned a few slides ago that calcium played a crucial role in muscle contraction. We'll take a look at that now. Remember, I said previously that ATP regulates the ability of myosin to bind to actin. Calcium regulates the ability of actin to bind to myosin. So when calcium is absent, the troponin holds the tropomyosin over the myosin binding sites. So we have the relaxed state here. The tropomyosin is overlying the myosin binding sites because calcium is not bound to the troponin complex. But when calcium becomes available, calcium binds to troponin C, which is the calcium binding component of troponin. Calcium is present, it's binding to the troponin C, causing the movement of the troponin. If you notice here, we have our troponin shifting laterally. It is shifted from this position to this position, exposing all of these myosin binding sites. And this, of course, is by exposing the binding sites on the myosin, for the myosin on the actin. It's going to allow cross-bridge formation, and it's going to allow the cross-bridge cycle to occur and contraction of the muscle. Now, we've been talking about muscle contraction. How does this happen? Well, we refer to it as excitation, contraction, coupling. Now, in a previous lecture on synaptic transmission, we talked about synaptic transmission at the neuromuscular junction. And if you remember, acetylcholine is released, binds to cholinergic receptors, specifically nicotinic receptors, causes a graded potential, which ultimately is going to cause activation of voltage-gated sodium channels, which is going to cause an action potential on the sarcoplasm membrane. It is that action potential we're referring to here from the excitation. It is the action potential in the muscle membrane itself is going to cause this cross-bridge cycle to start causing contraction. Now, why we refer to it this way is this excitation, contraction, coupling is just like when we talked about sensory systems. We talked about transduction, changing one type of energy into another. So in this case, when we talk about excitation, contraction, coupling, and we talk about this whenever we talk about muscles, we'll be talking about this in the heart as well. It's the electrical excitation, the electrical energy, the action potential gets converted into mechanical energy, that is contraction. And so, of course, dependent upon neural input from a motor neuron, because it is the release of the neurotransmitter that causes the action potential in the muscle membrane. And then, of course, the calcium release from the sarcoplasmic reticulum. We talked about how calcium, by binding to troponin, is going to alter the binding of the ability of actin to bind to myosin. That calcium is coming from the sarcoplasmic reticulum, so we'll talk about the details of how that occurs in a few slides. So the steps in the excitation, contraction, coupling. Well, we have the action potential produced in the sarcoplasmic membrane or the sarcolemma, and the action potential travels down a T-tubule. So let me advance to another slide here. We have our neuromuscular junction, have an action potential now traveling along the membrane of the muscle cell, and it comes down the T-tubule. T-tubule, remember, it's a transverse tubule. It traverses, cuts across the muscle fiber, hence the name T-tubule. So the action potential travels down the T-tubule. It's then going to do fairly complicated things on what we call DHP receptors and ryanodine receptors. Now, these are not shown in this slide. They're shown in the next, but they occur along the membrane of the T-tubule here. And let me jump ahead to the next slide. So we have here the DHP receptor and the ryanodine receptor. The DHP receptor is what's shown as a sort of goldish structure on the T-tubule membrane, and the ryanodine receptor is shown attached to it. The DHP receptor is actually a voltage sensor of the calcium tubule, and it's going to activate a calcium channel. So these are voltage-gated calcium channels, but they are named for the different subunits. So the DHP receptor, DHP stands for dihydropyridine, acts as the voltage sensor or the voltage gate that actually opens up the calcium channel, which is called the ryanodine receptors. Now, this is pretty frustrating terminology, but it's named for the two drugs, dihydropyridine and ryanodine, that were discovered and can be used in heart failure. Heart failure is when the heart muscle cannot contract forcefully enough to eject as much blood as it should. So these so-called receptors are named because they act as receptors for these drugs. But what these drugs are doing is altering the gating sensitivity of the calcium channel as well as the calcium channel itself. And in so doing, they're making more calcium available to cause the muscle to bind more forcefully. And that is why we have this terminology, because of these proteins as being drug targets. Basically, we then have calcium release from the endoplasmic reticulum. So I'm jumping ahead a couple of slides here. We have calcium that's stored in the endoplasmic reticulum that can't get out of the endoplasmic reticulum until the action potential activates the DHP receptor, which is physically linked to the calcium channel. It's going to open, and the calcium channel, remember, is also called the ryanodine receptor. It's going to open the calcium channel, allowing calcium to move from the sarcoplasmic reticulum into the intracellular space surrounding the sarcoplasmic reticulum. So then we have calcium concentration increasing in the intracellular fluid, and of course it's going to bind to troponin, shifting the tropomyosin, and it's going to allow the cross-bridge cycle to occur. So I'm going to skip this slide. Serves as sort of a transition between the two, and go to this one because this is going to have the greater detail that we need. So first of all, calcium channels open two ways. There's a voltage-gated opening, and there's a calcium-gated calcium channel opening. It's a bit of an odd thing compared to what we've talked about so far, but actually the calcium is also able to open the calcium channel itself. We'll talk about that in a moment. So we have these voltage-gated opening. This is due to the voltage change from the action potential affecting the DHP receptor. So the DHP receptor acts as a voltage sensor, and it is physically linked to the ryanodine receptor. The ryanodine receptor is the calcium channel. Excuse me. So when an action potential invades the T-tubule, causes a conformational change in the DHP receptor, changes its shape, pulling open the ryanodine receptor, allowing calcium to flow through. Now what gets kind of messy about this is this calcium-gated calcium channel. We'll talk about that now. So these calcium channels also can bind calcium, and in fact they have two calcium binding sites. One of them, it's a high affinity calcium binding site, and then another one that's a lower affinity calcium binding site. They are two distinct sites. The high affinity site is responsible for calcium-induced opening. So what happens is when we have an action potential coming down the T-tubule. Causes a conformational change in the DHP receptor, allowing some calcium to come out. That calcium then is going to bind to other sites that is, other than the DHP receptor, on this calcium channel. And it's going to cause the channel to open even more. And in fact, some of this calcium may go out to other calcium channels. We have calcium, let's say, coming out here, coming down and working on this calcium channel without even needing to engage the DHP receptor. This has a high affinity for calcium. It makes sense because at this point there's a low amount of calcium available. So in order to bind calcium, it's going to have a high attraction or high affinity to it. But we don't want too much calcium getting out. Remember, the sarcoplasmic reticulum is a way of maintaining a reservoir for calcium. So as calcium comes out, we're going to want to stop increased calcium flow so that we don't flood the cell with calcium. So we have calcium-induced closing. This is a different binding site, and it has a lower affinity for calcium. So perhaps it's located, in a different spot on the ryanodine receptor. We don't know the specific details of it yet. We just know the generalities of how it works. But this is a lower affinity calcium site, which makes sense. You don't want it to have the same high affinity as the calcium-induced opening site, because then it would close immediately. So you want a certain amount of calcium to come into the intracellular fluid and be allowed to interact with troponin and cause cross-bridge formation before you turn off the calcium flow. So if you have a site that has a low affinity for calcium, it's going to take a lot more calcium before you have binding to it. This is, again, due to the law of mass action. For those of you that are struggling with this idea, you really need to go back and review the material about the properties of proteins and their saturability and affinity and selectivity. Because you have to have sites that have different levels of affinity for them to be operated at two different time points, and those two different time points are correlated with different concentrations of calcium. So once we've got contraction, how do we terminate it? Well, the calcium has to leave the troponin, allowing the tropomyosin to go back over the myosin binding sites and prevent myosin from binding to the actin. So we remove calcium from the cytosol with what's called the SERCA. The SERCA is the abbreviation for the sarcoendoplasmic reticulum calcium ATPase. That is an ATPase, an enzyme that breaks down ATP as a fuel for activating a calcium pump. So the SERCA is a calcium pump that then is going to transport calcium from the intracellular fluid back into the sarcoplasmic reticulum. And then, of course, the muscle can't contract because there's not enough calcium available. So this is concluding the first segment of this lecture on the molecular aspects of muscle contraction. Now we're going to start moving to a little larger picture, a larger level of organization. We're going to be talking about how muscle fibers, motor units, and whole muscles are involved in contraction. First of all, when a muscle contracts, we refer to it as a twitch contraction, whether or not it's the muscle fiber, a motor unit, or the whole muscle that we're referring to. The twitch contraction is produced in a muscle fiber in response to a single action potential. It's all or nothing, just like an action potential. It's either there or it's not, and when it's there, it is there at its full magnitude. And as I said, it can be defined at three different levels: that of the single cell, the motor unit, or the whole muscle fiber. So we have an action potential. We're talking again excitation, contraction, coupling. Action potential preceding the contraction, and this contraction is what we refer to as a twitch. And the twitch is going to reach the same magnitude each time it is generated. There are phases of a twitch. First of all, there's a latent period. This is a time between the action potential and the onset of contraction. It's just a few milliseconds, but it's this area here when there is actually no muscle tension developing. And excitation, contraction, coupling is going on during this time. So the latent period is not a period of nothing happening. It's just a period in which there is no visible change in the muscle itself. The contraction phase, which goes on between the latent phase up to the peak of the muscle tension, it's still fairly brief, 10 to 100 milliseconds, and it's when cross-bridge cycling is going on. So this is, of course, when the actin-myosin binding is going on. And then a much more prolonged phase is the relaxation phase. And then this is the time where muscle tension is decreasing and calcium uptake is going on during this time. So even though we have these three phases, and in two of them, the latent period and relaxation phase, it looks like nothing is really happening. Very important things are going on. They're just not that easy to see. Now, as you've seen in your lab, if you've done the lab yet, there are two types of twitches: isometric twitches and isotonic twitches. As a way of refreshing our memory again, keep in mind that the contractile elements are the sarcomeres. And the sarcomeres, because they contain the actin and myosin, with the cross-bridge shortening, the sliding filaments, etc., are going to cause the muscle shortening, the development of muscle tension, the development of a twitch. The idea is that when a sarcomere shortens, it's not a gradual thing. It sort of sounded it the way I've been discussing it so far, but it shortens a certain amount, pretty much instantaneously. Also, the muscle contains what we call series elastic elements. Talked about it before. Titin is an elastic element, also connective tissue, the tendons. These all act like elastic tissue, and they exist in series. That is, one attached to the end of another. Again, for terminology, the force exerted by the muscle is what we refer to as tension. It's when the muscle is preparing to shorten and shortening. And that the force opposing the contraction, that is, the thing you're trying to move, is what we refer to as the load. The load makes it sound like it has to be something heavy, like lifting weights at the gym. Just picking up a pencil to write notes, you're lifting that load with your fingers. So let's look at these types of muscle twitch. Isometric term means that the length is constant. Think about meter stick, and iso means same. So we're maintaining the constant length of the muscle. In this case, the contractile elements are contracting, generating force, but that they have not generated tension that exceeds the load. The load exceeds the tension. So all we're doing is stretching our series elastic elements, stretching the titin, the tendons, the connective tissue. The muscle does not shorten. The load is not lifted. If you think about this, it kind of makes sense. You're getting ready to lift, especially a very large weight. As you grasp the weight with your hands and then tense up your arm muscles, until the time that you actually lift that load, you are beginning to contract muscles. It's just that the muscle contraction, the muscle tension, has not exceeded the force of the load, so you haven't lifted it, but you're clearly aware of developing some muscle tension. So the muscle tension can increase, but if we had a load attached here, in this case we don't, it's just we have a bar that doesn't allow the muscle to shorten any. We can still develop muscle tension. In isotonic, we're producing a constant tension. So the tone part refers to the tension, and the iso again means constant. So isotonic contraction, we maintain constant tension. The tension exceeds the load, so the load is lifted. So there's a sequence of events, especially if you go to lift a very heavy load. You have an isometric contraction that precedes the isotonic contraction. So how does the load affect the tension? Well, as we increase our loads here, going from 5 grams, 10 grams, 15 grams, up to 20 grams ultimately, we start out, the first 3 contractions are isotonic, that the muscle can shorten because it can generate a tension that exceeds the load. So again, isotonic contraction. The tension exceeds the load. So we have shortening of the load. You can see this because this lever is moving upward. It's being pulled up along with the load. And what is shown here is that we have muscle tension developing, And the muscle tension does not change any once it is matched that of the load. That is, it is matched 5 grams. In fact, it's obviously just a little bit more than 5 grams, so that it need not generate any more tension because the load is not changing. And then, of course, it relaxes. We increase the load. Tension is going to have to increase above before because we have a heavier load, but once it exceeds that 10 grams, it's going to maintain that same tension, pulling this 10 grams up and holding it up. Once we get up here to 20 grams, we finally achieved a point where we can no longer have an isotonic contraction. We have an isometric contraction. The muscle tension is going to develop, but in this case, we only develop 17 grams of tension, and it's a 20-gram load that we're trying to lift. So when the load exceeds the tension, the contraction becomes isometric. The muscle cannot change its length. So what normally happens? We're talking about things in pretty artificial circumstances so far. Well, first of all, there is always some purely isometric contractions occurring. Also, we can have purely isotonic contractions, but they are rare. Even if the load is constant, isometric contraction precedes isotonic phase of contraction. The isometric contraction continues, tension continues to increase until the tension exceeds the load. And then the isotonic contraction begins. So that's why we say a purely isotonic contraction is rare, because it is preceded by some isometric contraction. Once the muscle shortens, the tension remains constant. As soon as it exceeds the load, it does not naturally shorten any more than what is required to lift the load. The other thing, too, is load is generally not constant. When we talk about muscle fiber experiments, you go back to these other experiments, we're showing a muscle that's been taken out of a body. It's a muscle in isolation. And of course, muscles don't exist this way. They exist in limbs. They exist working around joints. So suppose as you flex your arm or extend your arm, either way, you're moving that limb around a joint, and the geometric change in that angle is changing the load. This is something that it talks about in a little bit of detail in your book. So generally, your load is not constant. So normal muscle activity is a little bit more complicated than what we talk about in terms of simply isometric and isotonic contractions. We can also produce graded muscle contractions. The grading, just as we talked about graded potentials, that is varying degrees, depends upon the amount of tension produced by each fiber and then the number of fibers that are contracting. So this is how, when you go to lift a heavier load, as you try to exert more and more force, you are producing a certain amount of tension by each fiber, but you're recruiting more muscle fibers into the contraction. So how do we determine the tension produced by individual muscle fibers? Well, the number of active cross bridges. And the more cross bridges, obviously, the greater the force. The number of cross bridges that are formed depends upon the frequency of stimulation. That is, in the natural state, what is the frequency of action potentials coming in from the motor neuron? What's the diameter of the muscle fiber, of the muscle cell itself? Obviously, a muscle fiber with greater diameter is going to generate more tension. A bigger muscle generates more tension than a smaller muscle, so the muscle fibers behave the same way. How the muscle fiber changes in length, we're going to talk about that in a little bit of detail. It turns out that you don't want a muscle too contracted or too stretched. If you exceed what's called an optimal length, it's going to alter the amount of tension you can develop. And then the extent of fatigue. Fatigue is a fairly complex in terms of the sources for it. We'll be talking about those in a few slides. So how does the frequency of stimulation affect it? Well, first of all, increases in frequency of the action potentials in the muscle fiber itself increases tension in two ways. And of course, a greater frequency of coming in from action potentials from the motor neuron is going to produce this greater frequency of action potentials within the muscle fiber. The first of the two ways is what's called tropé. It's a French word that as you have greater frequency of action potentials, keep in mind that the relaxation phase here of the muscle is a more prolonged event than is the contraction phase. And that's when we are pumping calcium back up with our circa. So if you increase the frequency and it's taking longer for calcium to be pumped back up than it is for the calcium to be released, you will end up producing a greater level of contraction after several action potentials have occurred, all of which have increased the total amount of calcium availability. The second way that you can increase tension by stimulation is summation, similar to what we saw in neurons, that an action potential lasts 2 milliseconds and the contraction lasts 10 to 200 milliseconds. Well, eventually, if you just have one contraction here, this one twitch, just like we saw for summation and neuronal discharge, if the second twitch is initiated before the first one is completely dissipated, you're going to add to that so that we have some summation in the third one, etc. So each subsequent one is going to temporally summate on the preceding ones, causing a greater level of force. This is what we refer to as tetany. And when the frequency increases so much that you actually can't see the relaxation phase at all, you have what's called complete or fused tetany, where you do not have this staircase effect on the top. It flattens out. So the cause of summation and tetanus, the amount of tension depends upon the amount of calcium bound to the troponin. At high frequencies, the release of calcium exceeds its uptake, so the amount of calcium increases in the cytosol. Eventually, you're going to saturate the system. Again, remember, proteins have saturability. So once all of the troponin is bound to calcium, the cross-bridge cycling is maxed out, and you have a maximum tetanic contraction. Fiber diameter, the force-generating capacity, is equals the inherent ability of the muscle to generate the force. The inherent ability of the muscle to generate the force is in part dependent upon how many myofibrils can be packed into that fiber. So as a diameter increases, you're increasing the total number of myofibrils that can exist within that space. So the number of cross bridges, of course, depends upon the number of myofibrils. And more cross bridges in a sarcomere produce greater force. Larger diameter fiber has more cross bridges. More sarcomeres in parallel, that is, if we have one sarcomere next to another one, so depending upon how the muscle fibers are stacked together as well. If you have a larger diameter muscle, bigger fibers are going to have more sarcomeres existing in parallel, generating more force. Now, the fiber length and the length-tension relationship, this is something that's a little bit complicated. But once you work on it for a few minutes, I think it'll make more sense. First of all, we have what we call an optimum length-tension relationship, and it is this flat green zone at the top of this curve. The length of the fiber at the onset of contraction affects the force generated. So what we're showing here is, keep in mind, this is our sarcomere in all of these cases here. Our sarcomere. If the sarcomere starts out being fairly contracted, so we notice here our actin filaments are actually bumping into each other. It's going to be that much harder to shorten it more because as those actin filaments are drawn toward each other, they're going to actually interfere with cross-bridge formation between the adjacent actin fiber. So once the muscle has been shortened to this point, where these two actin filaments are bumping into each other and beginning to overlap, it doesn't really show well here, but what we have is that this is one actin filament and this is another, and then I'll use a different color for our myosin. And we've got our cross bridges coming out. This area of overlap here is preventing a maximum number of cross bridges formation because it's binding to one or the other actin filament, but it can't bind to both of them. Or at least here, we're having a maximum number of cross bridges formation because each myosin head can fully bind to an actin molecule. Once the sarcomere has been stretched out to the point that the end of the actin molecules lines up with the last myosin head before this bare zone here, we're doing the opposite. Now, if we stretch it out more, we're pulling the actin away, and there are going to be some myosin heads that will not be able to form cross bridges. Ultimately, if we pull it out so far, it can't form any cross bridges. So in between here, we have a situation in which the actin molecules are just touching to being pulled apart enough to start losing overlap with cross bridges. It is the area in between that we call our optimum length, which produces what's called the optimum length tension relationship. Optimum tension is developed when the muscle is at the optimum length prior to contraction. So basically, having it stretched to this point and contracting it to this point. So some of the factors that are going to affect a whole muscle contraction. Remember, we're talking about the fact that twitch can exist at several levels. Obviously, our bodies work with whole muscles, not single fibers. So how do we describe that? Well, first of all, you have more muscle fibers contracting, therefore you're going to generate greater force. How does this happen? The process of recruitment. I mentioned it in passing on a previous slide. What we're doing is that we're stimulating more muscle fibers to contract over time. So more muscle fibers contracting is going to generate more force. So we have here stimulation of just motor unit X, which of course is this motor neuron, plus all of these brighter reddish colored fibers that it innervates. Contracting 5 fibers, we have obviously our tension development here, and then we've reached this isotonic state. If you then add more fibers to it, or activate another motor neuron, so in this case we're going to compare motor neuron X to motor neuron Y. Motor neuron Y activates 7 fibers, not 5, so that we have a greater level of contraction. If we now recruit 7 in addition to 5, we're going to have a total of 12 fibers. So we're going to have much greater force. In fact, we have a summation of the amount of tension or force developed by both of these. So activation of more motor neurons, activates more motor fibers, generates more tension. The muscles that we use for delicate movements, such as those on your finger, generally have very small motor units. So we have very few muscle fibers being activated in the motor unit. We don't generate much force, but this is partly what allows us to have our delicacy of movement. Muscles that we use for strength have large motor units. That is, that the motor neuron innervates many, many muscle fibers. So, of course, you're going to be contracting a lot more muscle fibers at one time, generating strength. That makes it more difficult, of course, to execute fine, delicate, and coordinated activity. Let's take a look at the motor unit size. The number of motor units vary in different muscles. Small muscles that we use for delicate movements have small motor units. That is, a motor neuron that innervates only a small number of motor fibers. Large muscles generally have large motor units which activate many, many motor fibers and therefore produce an enormous amount of strength. Fiber diameter, we've talked about already, varies with motor unit. So the diameter is small for the weaker movements. So small motor units generally have small diameter motor fibers. Large motor units generally have large diameter motor fibers because they're necessary for stronger movements. So the size of the motor unit and the fiber diameter are often related. Small motor units innervate small fibers. Large motor units innervate large fibers. This leads us to the concept of the size principle, that recruitment occurs over time. And the order of recruitment, that is when one muscle unit is recruited, occurs in a fairly predictable manner. And it is related to the size of the motor units. Small units generally are recruited first, whereas large units are recruited last. The mechanism of recruitment. Well, a large motor unit has a large motor neuronal cell body with large axon diameters. The size of the cell body is going to control how easy it is to depolarize the cell membrane of the neuron and cause an action potential to be discharged. Small motor units have small motor neuron cell bodies, small axon diameters. The larger neurons are harder to depolarize to threshold. Why? Well, because they have a larger membrane, a lot more leakage channels, so that there is a lot more loss of potassium. Therefore, it's harder to depolarize them to threshold. So it's going to require greater synaptic input, and small neurons be excited initially. They have a low input compared to the large neurons. So just graphically demonstrating what we showed before, a small motor unit versus a large one. Early tension development, it's the small motor neurons, small motor units, I should say, that are active first, then the larger ones, and subsequently the largest ones. And this is dictated in large part by the size of the cell body of the motor neuron itself. Now we need to move on to another aspect of muscle physiology, that is the types of muscle fibers. And there are two different ways of classifying muscle fiber type: by the speed of contraction or the source of the ATP, that is the source of the energy. So we talk about speed of contraction. We have fast twitch fibers and slow twitch fibers. Fast twitch fibers, of course, develop tension much more rapidly than slow twitch fibers. Different muscle fibers have different primary source of ATP. They can use glycolysis. They can use oxidative phosphorylation. They can store small amounts of ATP available at the onset of muscle activity, but it has to be replenished immediately. So depending upon where that ATP is coming from, it will affect the availability of ATP. We'll be talking about this in some detail in some subsequent slides. So with the two systems, there's a final classification system, which is a combination of these. We can have slow oxidative fibers, fast oxidative fibers, and fast glycolytic fibers. Let's take a look at sources of ATP. There are three sources. I have them circled here. They correspond to the list on this side. I have them listed in the order of use. So as soon as you start contracting muscle, first thing you do is you phosphorylate ADP with creatinine phosphate, which is what's going on here. So phosphorylation of ADP. We have creatine phosphate, which gets phosphorylated. Again, phosphorylation means we're taking ADP, we're adding a phosphate to it, which is going to give us ATP. So this process here is what we refer to as phosphorylation. And creatine phosphate is used as that source. Oxidative phosphorylation occurs in the mitochondria. Now, in this diagram, there's a great deal of detail that's hidden in this particular box. It just says oxidative phosphorylation. But this is going on in the mitochondria. For those of you that have had a good cell biology background, you probably know all those details. If you haven't had a strong cell biology background, you may want to go to one of the earlier chapters in the book. I think it's chapter 2 or 3 that talks about mitochondria and talks about oxidative phosphorylation. And then ultimately anaerobic glycolysis, which is going on here. Anaerobic meaning in the lack of oxygen. So we have, first of all, a very rapid energy source with creatine phosphate helping to produce ATP very, very quickly. But this is going to produce a very limited source of ATP. So it's going to be exhausted quickly. Then, that's why we need to breathe so much more during exercise. We use the oxygen for oxidative phosphorylation in the mitochondria. This is going to be producing enormous amounts of ATP for a longer period of time than what creatine phosphate was, but still not for an extended period of time. So this isn't going to keep someone running a marathon, for example. And we'll be talking about this in some detail when we do get into the end of the semester, where we talk about exercise and running marathons, etc. But eventually, we're going to have to go into anaerobic glycolysis. Anaerobic meaning that it's occurring in the absence of oxygen. Not that there is no oxygen available, but there's not enough oxygen available to drive the total ATP production that we need. So we undergo this process of anaerobic glycolysis. And again, these are presented in the order of use. So we're starting with creatine phosphate, then oxidative phosphorylation, and eventually with anaerobic glycolysis. So let's take a look at the creatine phosphate system. Creatine phosphate binds to ATP, and in the presence of this enzyme creatine kinase, remember a kinase engages in phosphorylation, so creatine kinase is going to be phosphorylating the ADP, producing ATP. According to the law of mass action, as we use up ATP, we're driving the reaction to the right. So as more and more of the ATP is used up, this chemical reaction is driven in this direction. So the creatine phosphate is the first source of ATP. It can provide 4 to 5 times the amount of ATP present in the cell at rest. So we have a certain amount of ATP that's just stored, and then the creatine phosphate is going to multiply that as this ATP is used. We're going to be producing more of it, but it's one step. While it occurs very rapidly, it's also used up very rapidly. This is one reason why it's the first step, but also fairly limited. Oxidative phosphorylation is the primary energy source during light to moderate exercise. It can only occur if oxygen is adequate, because it is an oxidative process. So aerobic exercises are all examples of oxidative phosphorylation. To maintain adequate oxygenation, we of course have to have an increase in ventilation, and which means of course a healthy pulmonary system. Increased heart rate and contraction of the heart, so we have to have a healthy cardiovascular system. And those blood vessels going to the skeletal muscle have to be able to dilate, that is, to stretch open more to allow more blood flow to get to that muscle, to provide more oxygen to the muscle to engage in oxidative phosphorylation. So the energy source for oxidative phosphorylation, well, first of all, there are muscle stores. We've talked in previous lectures about glycogen. Remember, glycogen is basically a storage form for carbohydrates, and it is a substrate for oxidative phosphorylation for up to about 30 minutes. So it's why it's limited to light to moderate levels of exercise. So glycogen get used up in about 30 minutes. Then glucose and fatty acids delivered to the muscle by the blood, and these dominate after 30 minutes. This glucose, of course, is coming from gluconeogenesis, which is in part breakdown of amino acids as well as some of the fatty acids as a way of producing new sources of glucose. This occurs largely in the liver. Now, anaerobic glycolysis, when oxygen supply to the muscle is limited. So this is during intense exercise. Or for that matter, even at moderate level of exercise, once we've gone well beyond the 30 minutes, it's becoming intense exercise because of the duration of it. becomes anaerobic, meaning that it's using very little oxygen. And at that point, anaerobic glycolysis is the primary source of ATP. It's fairly rapid. However, it only produces 2 ATP per glucose molecule. And, of course, there's a limited availability of glucose. This causes a buildup of lactic acid. This is largely what causes the burning sensation in muscle when you're exercising at a fairly intense level. Anaerobic glycolysis can provide limited duration of high-intensity exercise. But at some point, because of the limited production of ATP, an individual just has to back off on that level of exercise intensity. Now let's look at the classification of fast and slow fibers. Excuse me. Whether or not it's fast or slow depends upon the rate at which the ATPase and the myosin actually hydrolyzes the ATP. So ATP hydrolysis, or breakdown of ATP, is the rate-limiting step. A higher rate of hydrolysis means faster cross-bridge cycling, which means that it will be a fast-twitch fiber. So fast-twitch fibers have myosin with a fast ATPase activity. Slow fibers have myosin with a slow ATPase activity. Fast fibers contract 2 to 3 times faster than slow fibers. Fast fibers also relax more rapidly because the calcium ATPase, the circa, operates faster as well. The slow fiber contractions last approximately 10 times longer than fast, so there's a trade-off. The fast twitch fibers contract very, very rapidly, but they can't sustain that contraction for as long a time as the slow twitch fibers. Now we're trying to bring it all together in terms of these classifications. So if we're classifying based on primary source of energy, we have either oxidative or glycolytic fibers. That is oxidative phosphorylation or anaerobic glycolysis. Oxidative fibers, the primary energy is through oxidative phosphorylation. These cells have many, many mitochondria. They have a lot of myoglobin, so that they are much redder in appearance. They're generally fairly small. This makes sense. If they're going to have a large dependency upon oxygen, they have to have a small diameter so that oxygen can diffuse very readily from the blood supply throughout the cell. And they are resistant to fatigue. Glycolytic fibers, the primary energy is through anaerobic glycolysis. They have fewer mitochondria. Of course, the mitochondria are part of oxidative phosphorylation. So if you have fewer mitochondria, you wouldn't expect a cell to be involved in oxidative phosphorylation. Conversely, they have many glycolytic enzymes. They have a lot of molecules of enzymes that engage in glycolysis. They have high glycogen stores. It makes sense. Glycolysis is the breakdown of glycogen, so they're obviously going to have a large storage of glycogen to support the glycolysis. They use very little oxygen. They are anaerobic. Therefore, they can be larger, because they don't need as much oxygen diffusion throughout the fiber as you have in the oxidative cells. But they're fairly quick to fatigue. So if you put it all together, you have 3 skeletal muscle fiber types: slow oxidative, fast oxidative, and fast glycolytic. And we'll look at their properties now. Slow oxidative fibers have a low myosin ATPase activity, but they have a high oxidative capacity. They're aerobic. A lot of mitochondria, rich blood supply. They are red because they have a very high concentration of myoglobin. Myoglobin is like hemoglobin, but it's what exists in the muscle, and it's a form of an ability to store oxygen in the muscle. So not only are these cells getting a high oxygen delivery because they are small diameter and have a fairly high blood flow, but they also have an enormous capacity to store oxygen. But they develop fairly small amount of tension because they have a small diameter. Of course, that smallness, as I said, produces a small diffusion barrier, which is facilitating delivery of oxygen. But overall, because they have a small diameter, can produce a small amount of tension, they are going to be fatiguing slowly, just they're not going to have a great deal of force generation. And they're fatiguing slowly because, of course, they have a large oxygen supply as well as this small diffusion barrier. Oxygen is going to get from the bloodstream into the muscle fiber that much more easily. The fast glycolytic fibers have a high myosin ATPase activity, a high glycolytic activity. They have a lot of glycogen storage, a lot of enzymes capable of breaking that glycogen down. But they don't have any myoglobin. They're not dependent upon oxygen, so they have no storage capacity. So myoglobin causes the red coloration, so if there's no myoglobin, glycolytic fibers tend to be white. Generally large diameter, they develop a fair amount of tension, but of course glycolysis does not produce as much ATP as does oxidative phosphorylation, so they fatigue fairly rapidly. Fast oxidative fibers, kind of a combination of the two. They have a high myosin ATPase activity, high oxidative capacity. They're aerobic. They have myoglobin because, of course, they're oxidative. They're slow to fatigue, but they're more rapid than the slow oxidative fibers. And they are intermediate diameter between the other two fiber types. Excuse me. So how are these distributed in a muscle? Well, first of all, one muscle is a mixture of fiber types. And the proportions vary depending upon the muscle function. An example, postural muscles, that is those that are involved in your back and your pelvis, have more slow oxidative capacity. They're not. They're necessary for a low-level, sustained degree of contraction to keep your posture upright. In a single motor unit, all muscle fibers are of the same type, but the type depends upon what kind of innervation, that is, what kind of motor neuron you get. And the metabolic patterns can change, but the speed cannot. So a fast oxidative fiber can become a fast glycolytic fiber, or vice versa, but the speed with which the muscle develops tension will not change. The muscle cells, muscle fiber types recruit in a specific sequence. The slow oxidative fibers are recruited first, followed by the fast oxidative fibers, and eventually by the fast glycolytic fibers. Now, as I mentioned several slides ago, quite a few in fact, fatigue is a fairly complicated thing. First of all, it's defined as the decline in the muscle's ability to maintain a constant force during repetitive stimulation. There are numerous causes. In high-intensity exercises, there's a buildup of lactic acid. This, of course, is what causes, as I said before, the burning sensation in your muscle. It can lead to severe muscle cramps. When the muscle is contracting, it's compressing blood vessels, which, of course, is limiting blood flow, which, of course, is going to decrease the amount of oxygen available to the muscle fibers that need it. Also, if the frequency of input from the motor neuron is fast enough, you can actually deplete acetylcholine. That's what's called neuromuscular fatigue. Less neurotransmitter means it's harder to generate an action potential in the muscle fiber itself. In low-intensity exercises, it's the depletion of energy reserves is the main cause of fatigue. Other possibilities, well, buildup of inorganic phosphates in the tissue, changes in ion distribution, that is sodium, potassium, other ions as well. And then ultimately what's called central fatigue. This is psychological fatigue. And it's not fully understood what controls it. It is thought that some of these other causes, that there may be some sort of sensors in the muscle that are detecting these other changes that we have here, and that this is being actually perceived as psychological fatigue. But clearly, the first three are very important in terms of actual physiologic limitations on the ability to produce continued muscle contraction and leading to fatigue. It can also have adaptation to use. Now, this can mean, of course, the effect of training where you actually enhance muscle function, but it also can be the effect of detraining when somebody is not actively undergoing training. Muscle rapidly becomes less effective in its ability to contract. So, skeletal muscle adapts to use. There is no cell division. What you do is you change muscle size due to a change in the size of the individual cells. You have hypertrophy. If muscle isn't used, you have what's called disuse atrophy. In this case, you actually decrease the size of the muscle fiber. You actually lose myofibrils. So we don't have more or fewer cells. We have more or fewer myofibrils, that is the contractile proteins. So we have atrophy due to disuse. You can also have atrophy due to denervation. If the motor neuron has been damaged or destroyed, you don't have any excitation, so therefore there will be atrophy due to lack of use, just as we have with disuse atrophy. You can have hypertrophy. This is an increase in size. In this case, we're increasing the number of myofibrils, which is increasing the size of the individual cells. And with that, obviously, there's going to be an increased production of actin and myosin in order to increase the number of myofibrils. Adaptations in aerobic exercise. Well, endurance exercises are aerobic exercise. Basically, low to moderate intensity of exercise can be sustained for a fair amount of time. So running, for example, bicycling, rowing, unlike something like weightlifting, which is more of a resistance exercise, it's not aerobic because the skeletal muscle in contracting is limiting blood flow to the skeletal muscle itself. But basically, the adaptation that occurs in aerobic exercise is that we're going to increase the oxidative capacity of the muscle. We're going to develop more mitochondria. We're going to increase the blood supply. You actually get more capillaries developing. And a decrease in the diameter of the muscle, which is going to, of course, facilitate the diffusion, or the muscle fiber, I should say, which will decrease the diffusion barrier, or increase the ease with which oxygen is getting from the blood supply throughout the cytoplasm of the muscle fiber. What happens in anaerobic exercise? Well, these are high-intensity exercises that can't be maintained. You're going to increase the ability of the muscle to generate more tension, doing this by increasing the amount of actin and myosin, the number of myofibrils, and the diameter of the muscle fiber, and increasing the number of glycolytic enzymes, that is, the number of molecules of enzyme that are available, and decreasing the oxidative capacity. This makes sense. If we're increasing the diameter of the muscle fiber, we have to decrease its oxidative capacity because a larger muscle fiber is going to be harder for oxygen to diffuse readily from the bloodstream throughout the cytoplasm. So now we're going to have to talk about the control of skeletal muscle activity. That is, what happens in a real live human being with an intact limbs. So muscle activity across joints and the receptors, that is, sensory receptors that are necessary for coordinating muscle activity. First of all, muscles can only pull. A muscle, when it contracts, it shortens. So it's going to pull. We can actually extend a limb by pulling a muscle that causes the limb to move in the opposite direction as when the muscle was flexed. So muscles have antagonistic action. In the lower arm, flexion of the lower arm is contraction of the biceps, but relaxation of the triceps. Whereas extension of the lower arm is the opposite pattern, contraction of the triceps and relaxation of the biceps. Again, keeping in mind that muscles can only pull, not push. So the limb pushes because we've altered the sequence of these antagonistic muscle pairs. Back in sensory system, we talked about proprioceptors, and that proprioceptors were somatosensory receptors that give us information about our body, but that we are not conscious of it. We do not perceive input from proprioceptors. The main types that we're going to talk about now are skeletal muscle receptors. There are two types, what are called the muscle spindle and the Golgi tendon organ. The muscle spindle itself consists of two components, what's called the type 1A and type 2 afferents. And the Golgi tendon organ consists of a type 1B afferent. So it's easy to get this confused because there's some overlap here. A type 1 afferent can be either a muscle spindle or Golgi tendon, depending upon if it's a 1A or a 1B. What's more important is that we remember these specific names, the muscle spindle and the Golgi tendon organ, and then as we get into the discussion of their function, that you understand that as well. Before we do that, we have to talk about intrafusal versus extrafusal fibers. The muscle spindle itself, so this is going back to the muscle spindle that we're talking about here, which is this, let me change color, this spindle-shaped structure that is embedded in the muscle. There are two types of fibers within the muscle spindle, extrafusal fibers and intrafusal fibers. We'll talk about the intrafusal first. The intrafusal fibers are actually, as the name suggests, within the spindle. Fusal is another way of saying spindle-shaped. So these are within the spindle, and they are contractile, but they do not contribute to the whole tension developed by the whole muscle. They do not contract or cause the muscle tension develop. What they do is they contract within the muscle spindle itself to help sustain the sensitivity of this muscle spindle. So they adjust the sensitivity of the muscle spindle to stretch, and they are innervated by what's called gamma motor neurons. The extrafusal fibers are the contractile cells of the muscles themselves. So these are the muscle cells we've been talking about throughout this whole lecture up until the time I mentioned intrafusal fibers. These are contractile cells of the muscle. They are responsible for the muscle tension, for the muscle contraction, and they are innervated by alpha motor neurons. So that's the thing you need to keep in mind, alpha motor neurons versus gamma motor neurons and what their roles are. Within the muscle spindle, the intrafusal fibers we've mentioned, connective tissue, and sensory nerve endings. So the important part is actually this spiraling structure that is a sensory nerve ending. These detect changes in muscle length. Now, if you remember when I talked about mechanical gating of ion channels, I said there can be pressure-sensitive or pressure-gated ion channels. These are located, it was believed, along these nerve endings that are here on the in the muscle spindle, and as the muscle surround them, as the extrafusal muscle contracts, it's causing the pressure across the membrane of these sensory neurons to change, which is altering ion flow across them. There are two types of sensory endings. What's called the annulospiral ending, which is the one that they show here that looks like it is actually a spiral. And then what's called the flower spray ending, because it looks like a spread of flowers such as daisies, and these are indicated not too well in this diagram, but it's coming into this muscle spindle. The muscle spindle, the sensitivity can be changed by the action of the intrafusal fibers. The idea there is as the extrafusal fiber shortens, of course, the spindle is going to shorten as well. And one way of keeping the tension on the sensory nerve endings constant, or to allow it to adjust to the change in the extrafusal fiber length, is to change the length of the intrafusal fibers at the same time. So what kind of responses can you get from these muscle spindles? Well, muscle spindles detect muscle stretch. Their whole function is to maintain an adequate amount of muscle tension to keep you within the optimum length-tension relationship. So this is, on this lower panel here, representing action potentials as we stretch or contract the muscle. So as the muscle is stretched, and keep in mind muscle spindles detect muscle stretch, of course we have a very large number of action potentials being derived from this muscle spindle. As we relax the muscle, there is a smaller amount of action potentials because there is less stretch on this spindle than there was before, less stretch on the muscle. And when the muscle is fully contracted, of course, the muscle spindle is not being stretched at all. So there are very few action potentials. Now, I mentioned before the intrafusal fiber and how important it is. It's one way of stopping a contracted muscle from totally silencing the muscle spindle. And we'll talk a little bit more about that in a couple of slides here. So we have this alpha-gamma coactivation. It's referring to the gamma motor neuron and the alpha motor neuron. So the alpha motor neuron is coming in and causing the extrafusal fibers to contract. That, of course, is shortening the muscle, which would cause the spindle to become slack. That's why it's shown sort of as this floppy-looking flaccid membrane here. But when you activate the gamma motor neuron, which innervates the intrafusal fiber, the intrafusal fiber shortens, helping to stop the membrane on the sensory receptor from going completely slack. This is coordinated in complicated ways within the spinal cord, so that even though we're shortening the spindle itself, actively shortening it, it is not confused as this representing a shortening of extrafusal versus intrafusal. The two are coordinated together. But it's a way of helping to maintain the sensitivity of the spindle to stretch. Now, I mentioned at the outset about muscle spindles that they are important in terms of determining muscle stretch and helping to maintain the optimum length-tension relationship. They do not provide protection to the muscle from overcontraction. The Golgi tendon organ, however, does. Golgi tendon organ, first of all, has sensory capsules that are within the tendon, hence the name tendon organ. And stretch of the tendon, which means, of course, contraction of the entire muscle, is going to stretch the tendon and activate the Golgi tendon organ. This is going to reflexively inhibit the muscle via activation of the type 1B afferent. The type 1B afferent is the afferent neuron arising from the Golgi tendon organ. This is going to actually inhibit the alpha motor neuron coming into the muscle, and it's to protect against overcontraction of the muscle. Because when the muscle contracts forcefully enough to start stretching the tendon, there is a danger that it could actually tear the tendon off from the bone. So that by having the Golgi tendon organ activated, it's going to decrease the amount of muscle tension that's developed and preserve this attachment to the bone. So again, the Golgi tendon organ is protective to the muscle, whereas the muscle spindle organ is not. The muscle spindle organ allows the muscle to work more efficiently by maintaining the optimum length-tension relationship.